Dr. Meenu. Dr. Meenu, can you hear me?
Sir, Swarnajit Singh sir, recording is started.
All right, sir.
Meenu is a co-host and you are host.
All right. Okay.
Okay. So
Yes.
Meenu madam can share her presentation.
I cannot contact Meenu, madam. Dr. Meenu,
She is there. She is there.
but she's not answering.
No, some problem with her mic.
Oh,
Some problem, probably internet connection may be low. Problem with her mic.
okay. Dr. Meenu, please off on your
She
laptop.
is listening you. She is listening you.
Okay.
But her audio not started.
Okay. Okay. Dr. Meenu, you can off on your laptop or mobile. It will work.
I think she has not started either video or audio. So she has to start video and audio.
Yes. Yes. Okay, I'll contact her by telephone. 1 minute.
Yeah, okay. Are you there?
Sir, please guide Meenu Saraf how to start a mic.
Hi, hello.
Hello.
Good afternoon. Good afternoon, everybody.
Yes, you are audible. You are audible now.
Good afternoon, sir.
Good afternoon. Good afternoon.
Okay. So, Dr. Singh, Dr. Singh.
Hello, Dr. Meenu.
Oh, yeah, hello, yeah. So I hope I'm connected all now.
Yes, madam. Now
Hello,
we
Dr.
can hear
Meenu.
you. Madam can share her PPT.
Yeah, okay.
Yes.
Yeah, is that all right?
Yes, Dr. Meenu has joined now and we can see your...
Yeah.
Dr. Meenu, can you hear me?
Yeah, I can. Am I audible to everybody?
I cannot hear you.
Yes, ma'am. Now I would like to introduce you first before starting the presentation. Good afternoon to.
I
Yeah.
think Dr. Meenu Saraf is starting the talk.
Okay. Should I start first?
Yeah.
Dr. Deepika, should I start?
Yes, ma'am.
Oh,
Ma'am,
you're
can I give a
starting?
brief introduction?
Okay, please.
Yeah. So good afternoon to the one and all present here, our esteemed delegate, patrons, and all the participants. I extend a warm welcome to you all. Today we gather here for an exclusive presentation and discussion on microbes, powerhouse of soil. Firstly, I would like to congratulate our patrons, Dr. A.M. Deshmukh, President MBSI, for providing platform for wonderful talks and discussion. I also welcome Dr. Swaranjit Singh, President MBSI. He's a director at SAS Free Polyclinic, Perch, Mohali. His name is listed in top 2% global scientists. Dr. Swaranjit Singh was awarded a prize by the DGCSIR for 2 of his publications that were listed in 70 top publications of the CSIR in its 70th Foundation Day. He has contributed to the society and masses by spreading science to the remotest corners of the country. He's a chairman biodiversity and chairman pioneers in microbiology, served in MTCC and Gene Bank, now the first international depository authority in India, member in World Federation for Cultural Collections Committee, member at Task Force National Biodiversity Authority Chennai, awarded Loyola Environmental Award. His publications are high in impact journals and the articles are highly cited in the literatures with H index 55, I-10 index of 147, and approximately 16,500 citations. Now I would like to introduce our speaker of the day, that is Professor Meenu Saraf. She's the Director, University School of Sciences and Head of Department of Microbiology and Biotechnology, Gujarat University in Ahmedabad, India. Alumni of the Department of Microbiology, Barkatullah University, and obtained PhD in microbiology. Joined the Gujarat University as a lecturer in 1992 and professor in 2005 and is heading the department since 2015. Research interests includes restoration of marginal land degradation of landforms with microbes and influence of abiotic and biotic stress conditions in plant microbial interactions. Her special focus is in biofertilizers, biocontrol, biofortification, systematic resistance, biofuel production, role of endophytes and nitrogen cycle in agriculture, sponsored by national and international funding agencies. Developed industrial and academic collaborations with several universities across the globe to research in conceptual and molecular advances to understand microbe host, especially pertaining to plant-soil microbe interaction, which makes it a fascinating, challenging to unravel the mechanisms and genetics of plant microbes interactions. University of Aberdeen, Imperial College London, etc. She has supervised 16 PhD, currently training 8, and have produced 30 MPhil graduates. Currently, she has 3 postdoctorals working under her supervision in the laboratory. She is also member of editorial board of scientific reports. Also, she is chief editor of Gujarat University newsletters and Gujarat University scientific journals. Her journals have I-10 index of 56 and H index of 28. Recipient of Young Scientist Award from Indian Science Congress, Platinum Jubilee Award in 2019, and Fellowship of Shastri Indo-Canadian Institute under the SMP program to University of Waterloo of Canada. She was also awarded the Outstanding Women PGPR Researcher Award. Recently, in October 2022, she received the Weiser Award, that is Women Involved in Scientific Engineering Research, from Indo-German Science and Technology Center, Gurugram, India. Congratulations, ma'am, for so much of achievements. And I welcome you to please start your presentation.
Dr. Meenu, You're welcome today to
Thank you
give
so
your
much.
talk, and we are so happy to listen to you. So please go ahead.
Respected Singh sir, Deepika sir, all my senior professors, Dr. Deshmukh, the society, thank you all so much. First of all, I'm very grateful to all of you for all your wishes and for inviting me here today for this talk that I'm supposed to share my views with you, as I understand, regarding what little I know about the subject. Thank you for giving me such a generous introduction, Dr. Deepika. I'm really feeling very honored about it. Now, coming to the talk, today, as you have read in my resume, I'm more of a soil microbiologist. And I really believe that soil is the powerhouse and microbes are the powerhouse of the soil. Soil has all the rhizospheric and bulk soil rhizospheric bacteria. And it is here that we get the full range of the microbes that are available. So if we look at this closely, what is the soil? Because I'm saying that microbes are the powerhouse of the soil. So what is this soil? If we look above the ground, it is a very small part that is there. But below the ground, the rhizosphere that we say, the root system, is very, very much larger. And all the difference, if you look at the close-up, there are so many different cyanobacteria, etc., etc. The big amount of bacteria, fungi, actinomycetes, algae, mycorrhizal associations, that is all present in the soil. Even the smell that we have from the soil when there is rainfall, it is also associated with the microbes. So we are all in very close association with all of these kind of microbes. So if I'm saying this is where the microbes are, so what is the location of my army? This is the army that is giving us power. So what is the location of this army? So the location of this army is in the root system. Near the root system, the rhizosphere, that is the 5 millimeter space that we are having outside the root system and in the soil. And apart from the rhizosphere also, there is this rhizoplane and all the different type of, all the different structures of the root system, which is involved in the betterment of the plant when we are looking at the productivity. So again, I come back to my powerhouse. When I say powerhouse and microbes are the powerhouse, what are they doing inside? So they are involved in root exudates, structure and shape microbiomes. They trigger the diversity of beneficial and pathogenic microbes present. The rhizospheric bacterial community, which is present in a large amount, it is functioning in terms of direct and indirect plant growth promoting activity. It is also competing with residential flora of soil and pathogenic organisms. It is colonizing the root systems and it is leading to biofilm formations. Slowly and slowly, we are coming nearer to what exactly they are doing. Now, of course, there are many different ways of sensing them, various types of quorum sensing and quorum quenching measures. Of course, there are a lot of pathogens, all kinds, not only fungal. And we all know about phytopathogens and how they are involved. Most of them travel through the root system, through the soil. Then there are bacteria which are involved in the control of these pests. So they are root shields and they are responsible for not allowing the bacteria to get inside the root system. This could be by so many things, by presence of enzymes, peptides, antibiotics, siderophores, volatile compounds, biosurfactants, etc. Coming closer again, microbiomes, where are they? Where are they seated? The bulk and the rhizospheric soil. What is the difference? Bulk soil is the soil which is outside the root system, and rhizospheric soil is like we define the rhizosphere. So if you look at the R/S ratio, this is particularly true for the studies we did in wheat. Then we see how the bacteria, actinomycetes, fungi, protozoa, algae are located. The majority of the R/S ratio is bacteria, right? So now, if I'm giving so much power to my microbes, what is the function they are doing? So my talk today is focused on the role of these bacteria as a tool for ecosystem services. So I'm saying that it is a tool for ecosystem service. Now, if I have to define this ecosystem service again into detail, I will say that bacteria are having two major contributions. There could be provisional services and there could be regulatory services. A little more depth into these. What are the provisional services that the bacteria are providing? The provisional service that these bacteria provide in the soil are physical support, first of all. They contribute to the soil formation, contribute to the nutrient cycling and organic matter productions. They contribute in form of raw material productions, producing antimicrobial agents and enzymes. They contribute in being a growth medium, which mobilize nutrients from insoluble minerals to support plant growth. Now, what about the other kind of service? The regulating service. What is the ecosystem service these microbes are providing in form of regulating services? Nutrient cycling, recycling of waste and detoxification, carrying out the filtering of contaminants. It is a rich habitat for biodiversity, biological control of pests, weeds, and pathogens, and also carbon storage and regulation of greenhouse gas emissions. So these are the major services they are providing. Now, coming still closer to the rhizosphere, there is a tripartite association, the tripartite association between the plant, the soil, and the microbes, and it is interconnected with each other. So it is this association which is very important, and all the three are interrelated. All the three are dependent on each other. All three of them are responsible for each other's positivity or negativity, whatever we say. Plant characteristics, root systems, nutrient requirements, root growth, nutrient uptake, exudations, biomass, microbial diversity, all kind of fixations of NPK fixations, organic acid, inorganic compounds, etc. The composition of the soil, the physical and the chemical constituents of the soil, pH, texture, nutrients, all of them are interrelated and they are in turn responsible for each other also. So now, as we are going so much into depth one by one, we will also see that there are water soluble compounds, there are amino acids, there are lectins, there are sugars, organic acids, which are food. Looking more closely into the rhizospheric biology, we will see that these interchanges are so much relevant to life there on the rhizosphere, and through the rhizosphere, the plant and what is happening to the plant. So higher the levels, the greater the chemical interactions, the greater the crosstalk between microbes, the pattern of root exudation, the biofilm formation. All these are responsible for the health of the plant and the ecosystem. Now, if we look a step back, the discovery of agriculture was the first big step towards a civilized life, so stated by Arthur Keith. So we all have either seen or read about it, depending on our age, of course, that there is a green revolution. There was a green revolution. Now, this green revolution happened because there were high-yielding varieties. There were chemical pesticides, which were used abundantly, pesticides, fertilizers, and irrigation potential increased. Use of farm machinery, agricultural loans, all of them increased. What happened? There was a green revolution. We all, India was no longer dependent on other countries, but we had become self-sufficient, and then we also became important because we started exporting the food grain. But then slowly what happened? Okay. All of this led to soil health degradation. The excessive use of chemical fertilizers led to a very reduced microbial activity. These microbes were non-target. I'm not talking of the phytopathogens or the ones which were desired, but the ones which were simply there and could no longer survive the onslaught of the fertilizer and pesticides. Excessive usage of macronutrients like nitrogen, phosphorus led to the development of acidic and alkaline soils. Also, the salinity increased because of ingress of water. The groundwater became contaminated. All of these problems related in big amount of pollution, water, soil pollution, loss of microorganisms, and resulting in an overall reduction in soil fertility because of so many things that we have said. So, what to do now? That is the question. There is low productivity, there is an increase in the population. There was a green revolution which led us to have some kind of benefits for one and all, enhanced use of fertilizers, crop plants, but there was also high incidence of pest diseases, and there were alarming levels of chemical residues in the food and soil. All of them posed a big health hazard and led to soil degradation. So now we understand that why we had to look at soil health, why we had to look at soil health and improve the soil health for the benefit of the plant. So, as I said, this is a tripartite association, and under the ground, what is happening is reflecting on the ground. So, exploring these microorganisms by unraveling their possible relations with plants has launched this new journey for not only for me, but for so many of us. For one way or the other, where we are working with biocontrol, with plant nutrition, plant pathogenic microbes, and phytostimulation, besides so many other things. So, there are stresses, there are biotic stresses. Biotic stresses caused by phytopathogens, maybe bacteria, fungi, virus, nematode, and insects. There are abiotic stresses because of drought, temperature, increase or decrease, whatever, salinity, acidity, sodic soils. And there is this plant growth promoting rhizobacteria, which is there in the soil, which is responsible for enhanced stress tolerance because of their phytohormone production, siderophore production, biocontrol activities, and induced systemic resistance. There is nutrient availability and uptake, nitrogen fixation, nutrient uptakes, solubilizations of phosphate and potassium. So all of these have to be balanced and used somewhere. So with this background, I would just like to quickly discuss what we are actually doing at our lab at the Gujarat University. I have divided them into certain projects. Like one of the projects that we are actively looking into is the micronutrient deficiency. So this is a kind of hidden hunger. We say we are having food, but the food is not giving us all the essential microelements. Micronutrient deficiencies are all over the country, whatever type of soil. If we are looking closely at it, then 43% soils are zinc deficient, 12% soils are iron deficient, 5.4% soils are copper deficient, 18.3% are boron deficient. And these deficiencies are important because ultimately they reflect into the plant. So it is not only the food security that we are looking at, we are also looking at the nutritional security. Is it giving us all the micronutrients that we desire to have? So the estimates today say that 7 billion people are suffering from malnutrition. Malnutrition is the kind of hidden hunger, anemic population, zinc deficiencies, etc. So zinc is very important, and zinc deficiency is spread worldwide. See in this picture that there is an orange thing and there is a green color thing. The orange one reflects widespread deficiency of zinc. So major part of India, as you see, has this deficiency. Otherwise, even medium deficiency is probably there in most parts of the world. And this is specifically called the hidden hunger because largely human physiology is dependent on zinc in a lot of cellular functions, and lot of as cofactors, as enzymes, etc. Similarly, iron. 60% of the world's population is iron deficient. Now, if you look at it closely, again, India comes with is iron deficient. It has severe iron deficiency, right? So many parts of the world also have deficiency, but we really have a lot of iron deficiency, and we believe that this should not happen because iron is a very important part of hemoglobin, and this would lead to other complications, diseases, and disorders. So what should we do now? How is biofortification possible through PGPRs? So PGPRs are in the soil, they are in close vicinity of the rhizosphere, and they are having the ability to chelate the metal ion present. So they are able to mineralize many of these metals into soluble forms and help in the uptake. That is what was seen. This is because of their ability of siderophore production. So if we look at the scheme, how it was designed, isolation of metal solubilizing isolates from the rhizosphere, the qualitative and quantitative analysis, what is the type of metal, ions and all, etc. How it is coming into the form where it is able to solubilize, and is it having any other role to play? Is it going to do the plant growth promotion apart from just the solubilization of the metal or the zinc? And ultimately, how this is going to be available in the plant. So, simple distribution, lot of diversity studies, lot of different type of mediums were used, and slowly we're able to come down to certain important things, like 87 isolates were obtained from different medium and different type of zinc compounds were studied, like sulfides and oxides and phosphates. And zinc solubilization was studied in simple laboratory conditions, where see, this is the picture which is showing us how the solubilization of zinc can be seen, and we can select the microbes which are having very good property of the zinc solubilization. Right? So not only this, so we are just not happy with simply just isolating them and seeing a clearing zone and, you know, wow, we've done it and this is it. But what is it? This is not some kind of magic happening. If it is happening, there has to be a base to it. And if we know the base to it, then we can talk about the improvements. And there is where our science comes. So we have seen that, okay, this is happening, it is there, but how it is happening? There is also this production of organic acids, which could be estimated by FPIR and HPLC methods, and different metabolites, zinc uptake genes, ZNU-A, ZNU-B, ZNU-C, etc. can be studied. Study of zinc ABC transporter genes and their structural analysis. And finally, the molecules which are behind the molecular docking and dynamic is studied. So all of them give us a very clear picture. Now, here I am just talking of zinc, but in the lab, we have also done iron, we have also done selenium along these same parameters. So pictures speak for themselves. So the field experiments of wheat with different varieties of wheat show the differences in the control and the treatments of wheat. So with PGPR, without PGPR, you know, even if you pass through the field, you can see the difference on both sides. And this is not only an attractive way to reduce the chemical fertilizers, but it is also an important way of crop biofortification. So now the question comes, how much of crop were we able to biofortify? So available zinc and iron, both are there in this graph of wheat. And the Exiguobacterium or renticum showed an increase in the seed, zinc and iron. So 18.2 ppm and 24.64 ppm of zinc and iron was increased. Now this is also very much evident. This is also very much evident in our graph that we are seeing, that how much increase is possible. Now, this led to some of the publications, biofortification of Triticum aestivium through inoculation of zinc solubilization bacteria, and importance of zinc, how to conquer zinc malnutrition. With this, we come to our second project about the integrated nutrient management. So these are small projects that the PhD scholars are doing. I am really not picking up test tubes or going into the field and doing a lot of work. I am more on just looking at what is happening. And students come with brilliant ideas. Some of them go through it. Some of them find it difficult. But I would just like to support them and say that they are doing some wonderful work. So this idea of the sustainable agricultural production involves use of more natural resources. So I&M is an important concept because all the nutrients are available in the soil. And we have to learn to integrate the use of all these natural and man-made sources of plants, nutrients, so that crop productivity increases and it increases in a sustainable way, which is very important for us today to understand that we have to maintain this productivity for future generations too. Here we developed a novel concept of the rhizobacterial consortium. So up till now, we were working with single isolates. But later we realized that two or more organisms, if they are associated through a physical attachment, exchange of primary or the secondary metabolites and behavior adaptations, right? So consortia are more resilient to environmental stress. They are having better growth constants. They are having a broader habitat range. Their range of population is higher, and they can mineralize a lot of nutrients. So, with this specific example, we used some of the very excellent bacteria like Brevibacillus, Enterobacter, Acinetobacter, Micrococcus, and the Bacillus species. And we went out to create their consortium, which is popularly known as the PGPC. So it was not co-inoculation, but it was a consortia development where the population of each was monitored. Different treatments were cultivated, T1, T2, T3, depending on which were compatible with each other, which could support the growth of each other for an extended period of time. Just not in 24 or 48 hours, but for a long period of time, they were labeled, they were marked, and their survival was checked. And monospecies and mixed species consortia all was then studied. With relation to growth profile, IAA production or phytokernel production, the phosphate solubilization, siderophore productions, and the SECD productions, which are important for tolerating the stress conditions. So in all these cases, the growth curves for growth, for phosphate, for IA, you can see the difference. See, if you look at the graph here, IA production is maximum 60 μg per ml, whereas for the consortia, this is 120 μg per ml. It is almost double. So that was the development of consortia, and exceptionally results which were very high were obtained. Same goes for siderophores. You can see from the scale itself, 35 versus 90 amount of siderophore production μg per ml, how much it is differing. Or the activity, ACC deaminase activity, which is important because it was helping for the plant to tide over. Now, this group of results, we put it for the biofuel crop or for the Jatropha development. And when we were analyzing this, these are the nursery trials that we did of the Jatropha plant, different consortia, different plants, and we went on to multi-locational field trials, right, with these consortia, and we got excellent results. Right from the seedling vigor index, there was about a 75% increment in the seedling vigor index. Similarly, as in the lab, in the field also, we saw increase of all these vegetative parameters, 60 days. See, the result was very apparent from the initial stages itself. Now 60 days, because the plant is jatropha, the seed is very hard. It takes a very long time to germinate and start growing. But the result is very much apparent. So not only the plant, but the chlorophyll, etc., everything was very much increased. And finally, so this is the big plantation that we developed for the jatropha crop, and we got very good, excellent results. Soil sampling, as I said, was done. So it was not only the plant, but the soil, because here we are talking of integrated nutrient management. And that is why every part of the soil was analyzed: pH, salinity, electrical conductivity, soil organic carbon, available nitrogen, available phosphorus, calcium, magnesium, all parameters were tested. But of course, all of them did show good results. But what we were looking at actually was the biofuel development, or the jatropha seed development. And this increased beyond doubt. The oil content, the grain yield, of course, ranged from 300 gram per plant and uninoculated to maximum 742 gram per plant. These are the seeds that you see on the lower left-hand side and the upper right-hand corner of your screen. And this is quite phenomenal. From 300 grams in uninoculated to 742 in inoculated consortia plants. And even the size of the seed increased, so that there was an oil content increase from 35% to 52% in the treated ones. So this was a very big success, and we were very happy with it. This, of course, led, because I'm from academics, and I really like to have a lot of publications around me all the time. So do my PhD scholars. So some which appeared, which I brought here from plant nutrition and etc. Now we'll go on to another project which we are working on quickly. I hope I'm still visible and you can listen to me. Yes.
Yes, ma'am.
Oh, fine. Thank you. Because see, sometimes it feels funny talking to a screen if there is nobody on the other side. And I don't know, I have been, thrown out from the net, but so nice of you to tell me that you are still there. So anyway, we started another small project on the floriculture, and we thought from the review of literature that this could be because we could involve the phytohormones here, and this could give us a good insight. So we decided to do it with the carnation. This is the common carnation that is there, and it is very widely used in all kinds of decorations. So how does the IAA extracted from the bacteria help in the biotization of seeds and regulates it later? So first step was optimization of IAA production, because our results, our concept is always very important. Whenever we start a big series, a project or a big series of experiments, we would like to start with the concept. We would like to start with the concept and then go ahead with the review and build a hypothesis around it. The hypothesis may be 1, 2, 3, however, and then try to build on the hypothesis and which one works out and which one does not. So slowly we will start here. So because we always thought that this was our hypothesis of dealing with phytohormones, that IAA would be an important thing to go around with. So the first would be the IAA production and optimization. Different things were taken, and however better we could optimize, we did. The optimization was so good that in situ staining that we carried out. Now, this is an important method we developed. This was the same Salvosky method, but we did it with the plant roots. This was the first of the kind, and it was published in Bioprotocols internationally. And uninoculated plants and inoculated plants, which are showing the pink color, are having good amount of IAA. Now, another, pictures speak a lot for themselves. So I like bringing in a lot of pictures. These are the soil plugs, the pinched stem end before the IA treatment, the first picture, and then dip it into the IAA, which has been produced by T bacteria. You see the root initiation very soon. In negative control, there are no visible roots in the soil plug. But when there is a positive control, the synthetic IAA was also used, a positive control, a negative control, and our bacteria, then Providentia and Klebsiella. So you see an even distribution of roots. In Providentia, 90% of the surface of the root plug is showing good root development, and in Klebsiella, 80%. So these pictures do not require any other explanation. There were important takeaways of how we could go about it and how to do it. Then we also saw that the gene, the decarboxylase gene, was important because this was an initiator or the inducer of the IAA. So the ladder was done, forward, backward primers in both were done, and we were able to isolate the IPDC gene. Another interesting picture, which is showing us the depth of the root morphology, because we have titled this as biotization of roots, how the plasticity of the roots changes. So in soil, how it appears, the scale is for our reference here, just water, synthetic C is just synthetic IAA, then Providentia SDM9, and these are the pictures which are showing how much the root hairs are developing. So again, in treated plants, see how many and how long root hairs are there. There is of course a scale attached to it. With this treated plant, the root phenotypes go to a long length, and we can see how these are so different. And now this is the other one which we did, the Klebsiella one. Here you see the phenomenal difference. So I have brought these two for comparison because I wanted to point out this difference, which is not very visible otherwise. And this is the microscopic pictures which are showing this big difference. We also went along with the GFP tagging, and both showed us very good results, and we could see this apparent in the root system also. So we have to come to the depth, and again we have to enforce what we have been thinking and how we have been able to achieve it. We also went on to do the confocal microscopy. This picture was very much appreciated. The localized GFP tagged bacteria on the root tissue. This is the Klebsiella, and this was such a beautiful picture where you can see the localized bacteria on the root hairs, how much they are there, the green color, because it is GFP tagging. And not only that, we went on to see their view in the epidermal cells, the 3D view of the epidermal cells, and also the root ectoplasm, where we could see some of these bacteria. In comparison, the non-inoculated plant had no bacteria. See, these are all plain pictures and there are no bacteria which are there. So this was really completing the picture for us. And the important takeaways from this was that the IPDC gene was very much there. It could be induced. It was 1.64 kb or 1.7 kb for the second culture SDM81. It is a tryptophan dependent biosynthesis. Biotization was possible. They were able to modulate root plasticity, and they were both very good rhizoplane colonizers. And this point was important because this we were doing in floriculture. So this portion of the work we were doing with the Pune, and there were these big greenhouse plants, the big greenhouse setups which are there, which are doing only floriculture. So they were very impressed by it and they wanted to have these cultures. But things don't work out very well when we are actually going in for this industrial and technical types, and there are so many issues about it. So anyway, we are very happy with our publications which appeared in the Plant and Soil. Now, besides this, we did do a lot of work on chickpea and consortia, where, see, in comparison to control, the amount of chickpea production per plant did wonders when we were talking of the treatments possible. Elongation of root systems with control, these speak for themselves. We also went on to study the enhancement of growth in stress conditions. So all kind of saline soils, sodic soils, landfills, we were slowly able to restore. Or it is a slow process. When I say it is slow, that means it takes a long time. It is just not that I add microbes and the tomato plant and it gives me good results. No, it does not. It takes a long time because the soil slowly has to build up the microbial consortium. And this cannot just be done by adding microbes to the soil. This is a very gradual process in the sense that it first has to be established with grasses. It has to be mixed and started with grasses, and then slowly you can build it up and build it up to other agricultural crops, which are short term, like seasonal crops, which you say, and then maybe at a later stage an even higher level of, because biomass has to be first established in the soil. Only then you can see all kind of wonders happening. So marginal landforms where we saw the chickpea growth and it was working out very well. Everything is being monitored at all times, physical, chemical, everything. So basically, I would say we learned how to improve abiotic stress tolerance, produce seeds which are high in protein, iron, and zinc. So deficiency could be solved. It was showing increased resistance to diseases. With the health, as with human health, the same with the plant. Because if with the human health, a healthy person is not catching diseases very fast. So with the plant, the improvement of the plant health will definitely have a property that there will be a resistance to disease. Okay. So we went on to greater depth because every time we carry out a project, we get another bright idea. So this time the idea was that what is the role of diazotrophs in enhancing plant growth traits and how we can highlight their role in precision agriculture. So from the beginning, we've been talking of that there has been an abundant use of all kinds of chemical fertilizers and pesticides, and somehow we have to cut it down. If we have to cut it down, we have to narrow our approach. So plant microbe interactions are critical for the ecosystem functioning, and we have somehow to narrow down this approach. We have to make it more precise. So precision agriculture uses the data from numerous sources to improve agricultural yield and reduces the crop management measures, fertilizer inputs, irrigation management, pesticide use. That means it is more site specific. Ecological processes, they have to be governed. What is the soil health has to be thought of. So all this together is put as rhizospheric priming. So what is rhizosphere priming is you have to prime the rhizosphere according to so many factors, numerous sources and inputs, and then precisely work on it so that you are not wasting anything and it is sustainable. So a better name for this also could be site specific. So growers are able to take large fields and manage them as though they are a group of small fields. So misapplication of products is not there, and this will increase the efficiency. So in this direction, we chose the most important one, which we have been always talking of, nitrogen fixation, the role of diazotrophs in agriculture. So now we are all very much aware of this kind of cycle where nitrogen fixing bacteria are fixing in form of ammonia. There are ammonifying bacteria, there is ammonia nitrifying bacteria, and to close the cycle, there is this denitrifying bacteria. And nitrate and the nitrogenous organic compounds can be exported in the xylem to the shoot system. So we thought that let us focus exclusively on diazotropes and then look into the precision agriculture and how it works. All this was based on some important hypothesis. But first of all, a little more of the basics. NUE is the nitrogen uptake efficiency. Nitrogen uptake efficiency would be output versus input. Input of nitrogens would be in form of BNF, fertilizers, depositions, and manure. And output would be the harvested product. In between, there would be some runoff, there would be some leaching, there would be some gas loss. So, this is the basic concept that we first put together and we then pulled it into a hypothesis. So, we wanted to do the precision agriculture, and for this we chose barley and pea crops, monocot and dicot under nitrogen limitation conditions. And then we thought, let us study how it is going to work on. This was the basic plan of work that was done. Collection of soil samples from either of the plants, and we were able to isolate some cultures and then measure total nitrogen, the amino nitrogen and the nitrate nitrogen. Evaluation of plant growth promoting traits, because whatever, even if it is a very good diazotrope, it is not doing any other work, it is not going to work out for us. The drought detection assay, acetylene reduction for the nitrogen fixation, of course, and identification of the strain. This is just for the isolation part. Now we developed an hypothesis which was based on having NIF negative mutants. The NIF-S mutant, which is lacking biological nitrogen fixation as well as phytohormone production. And the plant inoculated with this mutant and untreated are showing us the same result. Then there was the ability, there is the second one where the plant is inoculated with positive strains of BNF and IA producing, and they have to show us some result. Then there is a third set which was plant inoculated only with biological nitrogen fixing activity strains and how it is going to work. This was one of the hypotheses, and to support this hypothesis we developed another one where we were doing the seed coating and working out all these details that you see on the right hand corner of your screen. The nitrogen mobilizing events during the plant growth. What is above ground, what is nitrogen accumulation, what is the root uptake of soil nitrogen, and root proliferation by the bacterial IAA. So both, every time we are now trying to correlate the nitrogenous activity with the activity of the auxin production. We had some good Klebsiella strains. Since we were always dealing with phytohormones, the IPDC sequence was fully decoded and amino acid sequence was deposited. And here, both activities were checked simultaneously as per our protocol. Physical parameters were studied. The chemical parameters were studied. Okay. And at every different intervals, 45 days, evaluation of physical parameters was continued, chemical was continued. Again, pictures speak from themselves, and control versus the strains. And the last one is the mutant, the NIF mutant. And we can see how they are all faring differently. And again, there is no competition, I think, between the SSM strain and SDM strain with the others. Now, this is the overall picture that we could find in this precision agriculture. Nitrogen usage efficiency, host plants nitrogen uptake efficiency is NUPE that you see in the center in the white. And nitrogen utilization efficiency is NUTE, nitrogen utilization efficiency. Different formulas were used for calculation. Above ground nitrogen and available soil nitrogen, the grain weight, and the available soil nitrogen. So these were the formulas that were used for calculations, and we can see the difference in the barley has a much greater NUE, NUPE and a NUTE efficiency than chickpea. As with barley and chickpea, NUPE was maximum in the SDM strain, but it was also showing the SSN1 strain was also having, but it was not the highest. So this was leading us to how much nitrogen when taken up is reflecting in the upper part of the plant and when it is not reflecting. This was what we were trying to do. So significant conclusions were there, like the Klebsiella, the isolate was Klebsiella quasi-pneumonia. And we could emphasize that Klebsiella can be used as an excellent tool to improve root architecture and the nitrogen usage efficiency. The wild type Klebsiella strains were test strains and they were used. And the nitrogen use efficiency was detected. Nitrogen mutants, nitrogenase negative mutants of M5A1 were used as negative control because they were not having any nitrogenase activity. And if we were able to use this formula for nitrogen usage efficiency, then overall we could lower the amount of nitrogen fertilizer used, and hence we could have this negative effect on the, prevent on the negative effect on the environment, right? So the role of 3 nitrogen fixing Klebsiella strains was studied, and barley and chickpea was studied as an example of monocot and dicot, and 8-fold and 3-fold increase in root surface was evident because they were all always good plant growth promoting, but some had no activity, some had good activity. The difference was in their nitrogenous activity and their phytohormone producing activity. So this, we could correlate all of them together and come to a very nice conclusion. A whole genome sequence was also done, and we were able to all submit it to the plants, which was also published, and there are 2 publications which are in progress. So I think I have taken up all my time in trying to discuss some of the things. Okay. I think I have come to the end of my presentation.
Okay. That was a very good presentation by you.
Yeah,
and it was
thank you.
very informative. We learned a lot of new things from your talk. And thank you so much for the nice talk. Now, I want to ask you a couple of questions.
Yes, sir, please. I'll be happy.
Yes, very good. Recently, you got a German award.
Yes, sir.
How did you feel when you got the award from Germany?
Oh, it was unbelievable, sir. I'm really so happy. I could not believe it. And really, it was quite a humbling experience for me because, sir, we actually don't think in terms of the awards and all. We just continue to do our work passionately, and we are happy to see the results and the pictures and the students who come out of it so nicely. So, well, sir, this was an award that was given by Indo-German Science and Technology Center, and this is an offshoot of DSC. And they identified 10 women from all over India, and they gave this award, which is termed as the Weiser Award, that is Women Involvement in Science and Engineering Research. So there were people from engineering background. I think there were 2 from the veterinary background, and there were 2 of us from the plant background. So, well, it felt very nice. And I'm really grateful to all my colleagues, my superiors, and my PhD scholars, sir, because they are the main who are doing all this important work.
We congratulate you very much for this award.
Thank you so much, sir.
And you used a new term, rhizosphere priming. Can you tell us something more about this rhizosphere priming?
Sir, actually, rhizosphere priming, when I say it is, we are not see any before when we talked of green revolution, we talked of high yielding varieties. And when we slowly talked of high yielding varieties, then overall we were giving a blanket of fertilizer, of pesticides, everything was happening like everybody was doing the same. But in this rhizospheric priming, it becomes very, very specific. If this particular small field has maybe some properties, it is deficient in some form of zinc, for example, or it is having zinc, but it is not mineralized. The plant is not able to take it. Then it can be treated differently. So in this way, we will not be wasting our resources. We will not be overthrowing our resources, so that what is happening is we are having a lot of pollution because groundwater water is affected. And this is coming back to us again and again. And we are seeing the disadvantages. There is an increase in cancers. Every part of our body is now showing this toxicity, which is coming from the plants. So how to stop it? So that is why this term has been coined. This is not coined by me. This is the rhizospheric priming, which has been coined so that people are able to look at it more closely at the rhizosphere and then give it that appropriate amount of nutrition or fertilizer or pesticide which is required.
Right.
Basically, we should all try to maintain the active microbial biomass, and that should be a perfect indicator.
It's a very nice term. I like the term, rhizosphere priming. Now, what is a co-inoculant and what is a consortium?
Sir, if we put two bacteria together, bacteria, fungi, bacteria, whatever, together, it is a co-inoculant, where we are not actually looking into their viability, we are not looking into their survival at a long period of time. We are just looking at it closely. We are thinking, okay, this is doing job, the nitrogen fixation very well, this is doing the ACC minus very well. So we just put it together and we hope that they will both survive and continue to do their job. But that does not happen. When we say that we are doing the co-inoculant, this is what is happening. But when we are doing it beforehand, we are doing this compatibility test that they are not eating each other up. They are not antagonists to each other, and they will let each other perform their work.
Right.
That is where we try to develop this consortia, and we use this word consortia, where both are living and doing the function they were put to do.
Yes, I understand. Do you preserve and conserve the microbes you isolate? Do you preserve them?
Yes, sir, we do.
With
In
yourself?
our lab, we do preserve, and we submit the gene sequences, and there also preservation is happening.
Are you submitting to some culture collection?
We are submitting to culture protection centers also, like to Chandigarh, Biotech Chandigarh. And
Okay.
also we are having our role in the department where, you know, like legacy of the department, we do continue to our.
Right. But once you change your interest,
Yeah.
your cultures will be lost. So
Yeah.
it is
Yeah.
always better to give it to some service collection.
Yes, sir, you are right.
Supposing you want to patent a strain, you
Yeah.
want to patent a process, you have to submit to a culture collection.
Yes, sir.
So I suggest to you
Yeah.
that the important consortium, nowadays you can also preserve the consortium as a whole.
Yes, sir.
Then you
And-
please deposit to a very good culture collection so that you don't lose your hard work.
Yes, sir.
Try
Yes, sir,
to-
I understand. Yeah.
Try to
We do
deposit
face
to
this
culture.
problem. Yes, sir. Because
Yes.
sometimes it happens.
Because, you know, once you change your interest, the cultures are lost.
Yes, sir. We do try our best to do it our way. But yeah, it happens. It does happen. And apart from the sequences that we have been depositing in NCBI collections,
Right.
we should be having a more thorough way. We have also been trying to talk to Professor A.K. Saxena about it.
Right, right.
To-
Yeah, that's a good idea. That's a good idea.
yeah.
Okay, anybody in the audience, they have any questions, you can please ask.
Yes, very good afternoon, ma'am. Very nice presentation and very relevant information you shared at your at the time of the presentation. One query is there
Thank
from
you.
my side. And like a lot of work is going on the nanoparticles. So
Yeah.
have you tried to convert the IEA into nanoparticles so that there will be more absorption with related to plants?
I have not started working with nanoparticles as such, or IA or anything else, because personally, I'm not very sure that we should be using nanoparticles in agriculture. See, what happens is the size is so small that it transmits through the cells. It does not have those biological barriers, are not barriers for. And in the long run, what is going to happen if we are going to eat these things which are having nanoparticles? That is the only reason. But apart from that, what I would also like to, what I would feel is that there are a lot of secondary metabolites, even without nanoparticles, which are doing their job. And maybe we should go more towards the natural things instead of developing nanoparticles, and use carriers of natural origin, which will probably do the same job.
Okay, ma'am, I'm talking about the nanoparticles that we are creating from the biopolymers. Because if you are tagging your IA with the biopolymers, definitely it is going to increase the surface area, and
Yeah.
that is how the absorption will increase. So yes.
No,
Okay.
I did not try it out yet. I did not try it out, but maybe yeah, if it is a biological based substrate, then it would be very good.
Okay,
Because we don't want to have any other chemical, any more chemical into the soil now.
okay, because recently I have started a work on doing generating the nanoparticles and tagging it and checking its activity with the PGPR.
Okay, very good.
Yeah. Also, I would
Yeah,
like to
I would like
highlight,
to-
I am also a student of Dr. D.K. Maheshwari.
Oh, how nice.
Very good. Very good. Okay.
Yeah,
Thank you,
great.
sir, and thank you, madam, for giving
Yeah,
this.
so Shreya, you are
question?
working there now?
Any other question, please? Anybody wants to ask question? Okay, I will ask you the last question, madam.
Yes, please, sir.
You know, the micronutrients are very important for crops.
Yes, sir.
Which are these micronutrients?
Sir, micronutrients would be zinc, iron, selenium, molybdenum, boron. So they are important for the crop, and they are also very important for us. Because
Right.
when we talk of malnutrition and when we talk of hidden hunger, it is, deficiencies which are more common. Now, every now and then we come across, even for ourselves, that B12 deficiency, vitamin D is missing. And all this is so much a part of the endophytic bacterial composition.
Right.
So not
right.
only for plants, I think for us, these micronutrient deficiencies are very important, and that is why it is called hidden hunger, because we are having enough to eat, but we don't have these specific things in our diet somehow.
Yeah. They are micronutrients.
Yeah, they are micronutrients, and we need them, but they are very important.
Yes. You know, in this context, I did some work on fortification of selenium into mushroom.
Okay.
And this work was done with collaboration with Tejo Prakash of Thapar University.
Very
And
nice.
this is a very important work because some population is deficient in selenium.
Yes, sir.
So we made a powder of mushroom, which was fortified with selenium.
Yeah, okay.
It is very useful for the deficient people in selenium.
So this could be then made into some other food items or something, or just direct?
Yes, it can be put in bread also, but people don't accept it, cannot accept it. So we put in mushroom. So the product is selling, it is selling a lot now.
Oh, very nice. It's very interesting. Yeah.
Yes.
Mushroom, yeah, it could be a good carrier anytime.
Yeah, it's a good carrier, mushroom.
Yeah.
So any other question, anybody? If no question, then I call upon Deepika. Deepika,
Yes, sir, I'm here.
yes. So I think we can conclude now,
Yes, sir.
please.
Okay. So I will, good afternoon, everyone, once again. It is such an honor for me to get the opportunity to thank all the dignitaries. I would like to express my gratitude to our esteemed speaker and delegate, Professor Meenu Saraf, for her presence and contribution to make this webinar a great success. I extend my gratitude to Dr. A.M. Deshmukh and Dr. Swaranjit Singh, sir, for providing immense support to make the webinar successful. And thank you all the participants for your patience, and thank you everyone once again. It was a wonderful presentation, ma'am.
Thank you. Thank you so much. Thank you so much, sir, for giving me this nice opportunity. Thank you, Dr. Deshmukh, Professor Singh. Thank you so much. And all the others who have taken time out for this presentation. I'm honored to have you all here.
Thank you very much. And I thank
Thank you.
Deepika. I thank dear Deepika so much for welcoming the speaker and giving the vote of thanks.
Yes,
Thank you,
thank
sir.
you. Thank you,
I
Deepika.
thank Deepika. Thank you.
Thank you. Thank you. Thank you.
Bye.
Yeah.